Okay, thank you very much. If we have the go-ahead, then I'm happy to introduce the next speaker in this split session. So Ruhee Dere from the Baylor College of Medicine in Texas, and she's going to be telling us about work that she's done looking at the inhibition of lysine demethylase 4A in terms of rescuing haploinsufficiency of 3P. So I hand over to you, Ruhee. You can share your screen.
Thank you, Ian. And okay, I hope you guys can see that. I'll go into presentation mode. So I'd like to begin by thanking the organizers for, of course, this opportunity and for organizing this tremendous VHL meeting, although it's virtual this year. Today I'm going to be telling you about lysine demethylase 4A. It's also called KDM4A. And this is a demethylase that we have identified as erasing the methyl mark on microtubules. And I think by modulating this demethylase, there's a potential strategy for rescuing some of the 3P haploinsufficiency. So needless to say, this is not something I need to address to this audience. But the important point that I wanted to make here was that the co-deletion of several tumor suppressors, VHL, SETD2, BAP1, PBRM1, sort of drive or contribute to renal cell carcinoma. Importantly for this talk, I'd like to focus on SETD2. It's an epigenetic regulator, and in about 90% of clear cell renal cell carcinoma, we see a monoallelic loss of this epigenetic regulator. Like I mentioned, SETD2 writes the trimethyl mark on histone H3, lysine 36. But more recently, I've been part of the team that discovered SETD2 also writes the trimethyl mark on tubulin of the microtubules. Mass spec has identified lysine 40 on alpha tubulin as the site for this trimethylation. And a methyl-specific antibody, which was, generated towards this particular epitope on tubulin, showed great mitotic spindle staining. And in cells where you've lost SETD2 or you don't see this methyl mark on the spindles, you see a lot of mitotic defects, micronuclei, lagging chromosomes, chromosome bridges, you name it. And of course, this contributes to genomic instability. So there's this very famous trifecta of writers, readers, erasers on chromatin, where they mark the histone tails. And this trifecta, which modulates or regulates or writes the histone code, we now believe is also working in the cytoplasm to write tubulin code. So I already told you about SETD2, and there's perhaps other writers, but we know SETD2 marks the lysine 40 site on microtubules. We've identified PBRM1 with a colleague of mine where we believe that this is the reader of this trimethyl mark on the microtubules. And we were more interested now in finding out if KDM4A could potentially be an eraser of this methyl mark. So why KDM4A? Well, KDM4A is the corresponding demethylase that erases this methyl mark on that's laid by SETD2 on chromatin. Enzymatically, it's a dioxygenase enzyme requiring both iron and alpha-ketoglutarate as cofactors. And this is data that we have generated, but this is pretty well established that KDM4A is not exclusively a nuclear protein, but in fact also localizes to the cytoplasm. We now have data to show that KDM4A binds methylated tubulin. So first I'm going to show you interaction with alpha tubulin. And this is in RPE1 cells where we've immunoprecipitated using an anti-alpha tubulin antibody. And we see a tagged KDM4A come down here. We've also done the reverse experiment where we've actually overexpressed EGFP tubulin. And here too, we're able to pull down KDM4A. We used this methyl-specific antibody for methylation on tubulin, and we were successfully able to also co-immunoprecipitate overexpress KDM4A. And we've done this using both a monoclonal antibody as well as a polyclonal for this methylation site on tubulin. To validate these data, the next experiment that we did was to IP KDM4A from HEK293 T cells and send it for mass spec analysis. And what we found by doing this is that tubulin 1A was hugely enriched in this KDM4A immunoprecipitate. So, of course, the most important question was KDM4A capable of erasing the methyl mark on tubulin? And the way we set up this experiment was to do an in vitro demethylase assay. Here we've used recombinant KDM4A, and what I've shown you first is a positive control, which was recombinant H3K36. And for our tubulin substrate, we've actually used bovine, this is purified tubulin from bovine brains, where we've run a Western and we've seen a decrease in methylation in the presence of KDM4A. This is quants from multiple experiments where we've used either bovine tubulin or we've used porcine tubulin. And in both cases, we see a reduction in the amount of methylation on tubulin. Now, to see if this was translated in vivo, we've overexpressed KDM4A in HEK293 T cells, or we've expressed a catalytically dead KDM4A. As one would expect, when you overexpress this mutant KDM4A, you see an increase in H3K36 prime methylation because you can no longer erase the methyl mark. And in looking at tubulin methylation, we also see a similar increase in tubulin methylation when we, in fact, lose catalytic activity of KDM4A. We went on to then look with our methyl tubulin specific antibody at the mitotic spindles. And what we've done here is we've taken MEPs where we have either our non-targeted controls or we've CRISPRed out KDM4A, and as you can tell, we see an increase in methylation both at the spindle as well as the chromatin, of course. We've also done the rescue experiment where we've re-expressed KDM4A, and now you see a decrease in methylation of the spindles and chromatin. One could ask, where is KDM4A localized? And we've used an antibody-based approach here where staining for KDM4A showed that it localized at the microtubule organizing centers during all phases of mitosis. And in order to see if this localization was dependent on SEDD2 methylation of tubulin, we used two different cell lines, the 786Os, which are SEDD2 proficient, where KDM4A nicely localizes at the microtubule organizing centers, and E5s, which are SEDD2 null. And in these cells too, we see good localization of KDM4A at the mitotic spindle, suggesting that this localization of KDM4A was independent of SEDD2 methylation of microtubules. So this led us to sort of lay out this hypothesis that if KDM4A opposed the methyl mark on microtubules laid by SEDD2, then could we in fact inhibit KDM4A and rescue some of the genomic defects associated with SEDD2 haploinsufficiency? And this is data that we recently published where if you lose two copies or both copies of SEDD2, you reduce your methylation on tubulin as well as chromatin. However, if you lose only one copy, or if you have mono-allelic loss of SEDD2, you see the decrease in tubulin methylation. However, your methylation on chromatin is unaffected. And this is looking at some of the genomic instability. Here we counted for micronuclei, and we saw a huge increase in micronuclei in these heterozygous MEPs. So the hypothesis then was pretty simple. If we have SEDD2 proficient cells, you would expect there to be methylation on the cytoskeleton as well as on the chromatin. However, in conditions where we had a monoallelic loss of SEDD2, could we now push the balance towards increasing cytoskeletal methylation by inhibiting the eraser? And that's exactly what we did. This is where we had METs, which were tamoxifen inducibly, which under tamoxifen control could inducibly use one allele of SEDD2. These are our controls, which were not treated with tamoxifen. And at the bottom panel, we have all the cells that were treated with tamoxifen, i.e. these were true monoallelic loss of SEDD2. Now, these cells, we had non-targeted controls. We had also CRISPRed out KDM4A, or we had rescued by re-expressing KDM4A. So let me walk you through this data. When we lose SETD2 or one copy of SETD2, you see an increase in micronuclear formation. And I've quanted that and plotted it here. You see an increase in micronuclei. However, in these same cells, now when you've lost KDM4A, you actually you reduce the percentage of micronucleated cells. But then when you re-express KDM4A, you're again increasing or driving a genomic instability. So this was truly exciting for multiple reasons, because this is not something that we would normally think of as a therapeutic strategy in renal cell carcinoma. So let me summarize first and tell you that showing KDM4A is in fact a cytoplasmic protein. It's binding alpha-tubulin. It demethylates microtubules, and it localizes to the spindle folds. So this was truly exciting because we already have a pharmacologic inhibitor of KDM4A that was developed at UT Southwestern. It's called GIBO4. And the idea here is that KDM4A activity could in fact be inhibited by using the small molecule GIBO4. The way this molecule works is that it disrupts oxygen binding in the catalytic domain of KDM4A. And so we're hoping that we can now sort of switch gears. This is all sort of fresh and unpublished data. We're hoping to now look at some preclinical models to see if we can in fact modulate KDM4A in mouse models. With that, I'd like to end and acknowledge the people that have worked on this project. We have the postdoctoral fellow, Dr. Ruhee Jangid, our research associate, Dr. Chaudhary, and of course, many, many collaborators here in Houston, as well as in Dallas and Michigan. For funding, I would like to thank the VHL Alliance that had the faith in me to fund some of my research, as well as the Gillis and Longbaugh Foundation, which has also funded part of this research. And with that, I'll be happy to take any questions.
Thank you very much. Really wonderful talk, and really, I think, opening up new avenues and certainly adding to a general concept that's emerging in the field of epigenetics, that these epigenetic regulators have many targets outside of chromatin and histones. So I think this is very nice. I have one question to start off with, and that is, what happens to KDM4A in CCRCC? Is it upregulated, downregulated? Are there different subsets of tumors?
So KDM4A is not really affected in renal cell carcinoma. So it's, it's fine. It's not overexpressed. And there are other cancers where, in fact, it is overexpressed and amplified, but that's not the case for renal cell carcinoma.
Okay. And maybe just to keep moving, we have two questions actually sent by Bernard Shem and Rachel Giles, and I wholeheartedly agree with these questions. It's close to all of our hearts. They're about primary cilium. So the questions are basically, do you see KDM4A in primary cilia or in basal bodies in ciliated renal cells? And how do you think, if at all, this might tie into the concept of that we have a VHL regulating primary cilia?
So that's a fantastic question, and I'm glad Rachel asked that, because although I presented this work, showing you the role of KDM4A in the mitotic spindle, the bottom line is that yes, in fact, it does localize to the basal body, and it does regulate cellulogenesis in very interesting fashion. So hopefully, I have time at some conference that we all go to in the future to talk about the primary cilia work. But yes, indeed, we do.
Wonderful. We look forward to hearing about that, hopefully in the near future.